Welcome to The Explainer. Today, we are diving into a complete forensic deconstruction of the AIPMT and Neet past papers from 2008 all the way to 2017. If you're prepping for this exam, or honestly, if you're just fascinated by how these massive, high-stakes standardized tests are actually built, you are exactly in the right place. Look, our goal today isn't just to review some biology. No, we are going to literally reverse engineer the exam itself. We're going to map out the exact architecture of the test, and specifically we'll be highlighting those highly repeated red danger zones and the green scoring topics you absolutely need to know. So it's the burning question, right? Every test prep student has probably stared at their ceiling asking this at 2 a.m. Where do these questions actually come from? Is there some secret vault? Are examiners just pulling obscure facts from high-level medical journals just to mess with us? Well, the reality is actually much more structured, highly predictable, and honestly, it's hiding right in plain sight. Okay, let's dive into this roadmap. We've got 5 quick stops. First, the blueprint of the exam. Second, decoding diagram-based questions. Third, the high-yield genetics and evolution zone. Fourth, the high-yield ecology and physiology zone. And finally, the NCRT advantage. All right, Section 1, Blueprint of the Exam. It's time to pull out our magnifying glass and look closely at the undisputed source code of this entire test, the NCERT Class 11 and 12 biology textbooks. Think of this as the fundamental architecture for the entire exam. You've got the foundational Class 11 units covering everything from diversity to human physiology. Then, the Class 12 units push into the more complex applications, things like genetics, biotechnology, and ecology. When we actually sat down and mapped a decade of past papers, clear patterns jumped right out at us. Examiners do not randomly select topics. They systematically distribute questions across these specific buckets. Some of these are what we call green topics, straightforward, memorization-heavy areas where you can rack up points fast. But others, they're red topics, conceptually dense areas where examiners just love to set traps. Moving right along to Section 2, Decoding Diagram-Based Questions. We all know the absolute pain of staring at a complex diagram, desperately trying to memorize every single arrow and label, right? But let's look at the anatomy of how an examiner actually takes that visual and turns it into a multiple-choice trap. Let's break down a classic mitochondria question from the 2011 paper. The examiners didn't just ask a general, open-ended question. They provided an image and asked students to match a specific labeled part with its precise function. So step 1 is simply identifying the smooth outer membrane. Step 2 is recognizing the inner membrane and its infoldings, the cristae. And step 3, this is the crucial one, making the conceptual leap to relate that specific inner structure to the enzymes of cellular respiration. Now, what's really interesting here is the strategic contrast between just reading a textbook fact and surviving a clever exam distractor. The actual biological fact is that the inner membrane forms the cristae. Simple enough. But the red zone trick the examiners threw into the options, they suggested the outer membrane splits or gives rise to the inner membrane. If you only kinda glossed over the visual while studying, well, that sounds plausible enough to make you second-guess yourself and pick the wrong answer. Let's deconstruct another classic visual, one that popped up repeatedly in 2009 and 2010, the standard electrocardiogram, or ECG. The test doesn't want you to be a fully trained cardiologist here. It just wants you to know the exact textbook correlations. Step 1, locate the P wave. Step 2, perfectly map that P wave to the depolarization or the contraction of both atria. It's a direct, one-to-one visual-to-text correlation, pulled straight out of the human physiology unit. Here's one more tricky one from the 2010 paper, focusing on the human eye. Notice how a super simple label becomes a crucial true or false separator. The basic fact is that the retina contains the photoreceptor cells, the rods and cones. But the exam trick? They threw in an option claiming the blind spot has only a few rods and cones. By definition, the blind spot has absolutely none. If you know your diagram labels with precision, you can instantly eliminate that distractor. Boom, moving on. Okay, on to Section 3, high-yield genetics and evolution. This right here is a massive red zone. It's conceptually heavy, mathematically tricky, and just generally tough. But interestingly, when we dig into the data, the historical context is tested almost as much as the biological mechanisms themselves. Tracing the timeline of these discoveries is incredibly high yield. In the 2015 and 2017 papers, knowing exactly when and how things happened was absolutely critical. For instance, examiners specifically tested the exact years of Mendel's hybridization experiments on pea plants, 1856 to 1863. They also love testing the progression from Watson and Crick's double helix discovery, all the way to the modern era, where the Human Genome Project literally gave birth to the entire field of bioinformatics. Knowing the history of biology isn't just trivia, it's treated as a core competency. But hey, it's not all history. Precision of vocabulary is absolutely non-negotiable. Take this term from the 2015 exam, the pleiotropic gene. The examiners offered trap options, saying it evolved during the Pliocene, or that it controls the trait only in combination with another gene. The exact, unyielding textbook definition is that a single pleiotropic gene controls multiple traits in an individual. In this section, close enough is simply wrong. And sometimes, an entire complex concept boils down to a single critical digit, 3. The messenger RNA-based sequences are read in combinations of 3. This triplet genetic code is exactly what dictates the placement of a single amino acid. Grasping this one simple rule basically unlocks a whole series of molecular biology questions for you. Let's pivot to Section 4, High-Yield Ecology and Physiology. Welcome to the green zone. These chapters are super dense with data, and the exams exploit this by using formats that are specifically designed to test your sheer volume of retention. Check out these respiratory capacities and volumes, tested directly back in 2010. It brilliantly illustrates how those infamous match-the-column questions are formulated. The examiner just takes 4 lines of data straight from the NCERT textbook, jumbles the numbers up, and asks you to match them back together. There is no deep concept to deduce here. It is pure, absolute data retention. Tidal volume is 500 milliliters. Residual is 1,200 milliliters. You either know the data matrix or you don't. We saw the exact same strategy deployed in the 2017 paper regarding human health. They hit students with a match-the-column question, pairing sexually transmitted diseases directly with their causative agents. Gonorrhea with Neisseria, syphilis with treponema, genital warts with HPV. Rote memorization of these pathogen disease tables is an absolute necessity if you want to secure these highly predictable, easy points. So, let's fire through 3 rapid-fire, punchy facts derived directly from the 2015 paper that you simply must have locked in your Mendel database. Ready? First up, babesiosis is caused by a sporozoan protozoan called Babesia. Fact 2. The filiform apparatus is a characteristic anatomical feature of synergids. This just reminds us how incredibly specific the examiners get when it comes to plant reproduction morphology. And finally, fact 3. Golden rice is a genetically modified crop plant meant specifically for the biosynthesis of vitamin A. So, the crucial point is, whether it's disease, plant anatomy, or biotechnology, these highly specific textbook facts are the exact ammunition you need to clear this exam efficiently. All right, Section 5, the NCERT advantage. We've dissected the diagrams, we've mapped out the history, and we've matched the data columns. It's time to bring all this evidence together. The forensic proof is undeniable at this point. The NCERT textbook isn't just a helpful guide, it is the definitive blueprint for success. It's the direct source for all complex diagram questions and those tricky distractors. It's the definitive origin of all the data used in match-the-column formats. It serves as the absolute non-negotiable baseline for terminology, like we saw earlier. If your definition differs from this book on exam day, your definition is marked wrong. And finally, it traces the specific historical timelines that the examiners consider mandatory knowledge. And this brilliantly illustrates just how deep the exam's reliance on this text really goes. This quote, defining reductionist biology, isn't buried in some dense, late-stage chapter. It's actually pulled from the introductory preface to Unit 3 in the Class 11 textbook. Think about that. Examiners are literally mining every single page, from the prefaces to the appendices, just to build this test. So, we've reversed engineered the mystery. The red zones and the green zones are completely mapped out. You now hold the exact blueprint of how the Neet biology section is constructed from the ground up. So the only question left for you is, how are you going to use this structured knowledge to build your score? Go hit the books, study smart, and I'll see you in the next explainer.